A couple words of explanation. I'll put this up here tomorrow too, but thinking about exam coming up next Monday. So our third exam will cover Chapter 8, Photosynthesis. And remember, the last section of that particular chapter will be some bonus questions. Chapter 13, we're only doing the first two sections. Chapter 14, we're also only doing the first two sections. If you start looking at old exams posted on the website, it gets a little confusing, because I think the 2013 exam, photosynthesis was on the second exam with respiration. So if you look at 2013, exam number 3, there's no photosynthesis, but then there's more RNA and protein synthesis stuff that we won't be covering. And so I wanted to put this up here and get this out there as a guide in terms of what you're going to be doing. So, this week in lab, we're going to be working with fluorescent organisms and transformation. This is a green fluorescent jellyfish. One of the questions you have to ask yourself in nature when there are some kind of bizarre appearances like this is, how come? How come an organism might produce something that allows it to fluoresce a green color? The reason that these particular organisms are able to produce a green color is because they produce a particular protein which has a unique three-dimensional shape. And the way this particular protein folds up, this GFP or green fluorescent protein, it is able to absorb blue and ultraviolet wavelengths of light. That excites electrons in those molecules, and then some light is re-emitted in the form of fluorescence, and the wavelength of light that is emitted is a green wavelength of light. So physically, the reason that these particular organisms are fluorescing is because they have a unique protein. Okay? Why do they have a unique protein? Because they know how to make it. They have instructions or genes that encode the information. Here is how you make a green fluorescent protein. You add this amino acid to this one, to this one, to this one, to this one. And then that information eventually gets turned into an RNA molecule, a messenger RNA. That messenger RNA will then move out into the cytoplasm and associate with ribosomes. And then amino acids will be hooked together, and the sequence of this particular polypeptide chain is dictated by the information that was in that green fluorescent protein gene. So it's very important to be able to distinguish between what a gene is and what a protein is. Okay? And it is the protein that is fluorescing, it is not the gene that is fluorescing. As you do your reading, I would like you to pay attention to and try to understand why organisms might make this green fluorescent protein, what advantage is there for an organism to have this particular gene. When we talk about gene expression, remember, involves turning information, genes, into tangible product, proteins. And one of the things that we're going to really stress as we go through gene expression is that expressing genes is expensive. Particularly, it requires a whole lot of energy to express a gene and make a protein. So one of the questions I'd like you to ponder and think about is, why are these jellyfish expending energy to make this green fluorescent protein? What might be the advantage of that particular phenotype of being able to fluoresce? Well, jellyfish can make this particular gene because they have the genetic information. This is the genetic code for the green fluorescent protein. Jellyfish have these genes. The organism that we're going to work with in lab, the bacteria E. coli, doesn't have this gene unless we give it to them. And so the purpose of this particular lab is to try to infect, if you like, these E. coli bacteria with a gene from another organism and see if they can acquire the ability to express that gene and make green fluorescent protein when they previously couldn't. That's transformation. Remember, that's what Griffith discovered, is the ability of genetic information from one cell to be taken up by another cell, and then hopefully that new cell will express that particular genetic information. Okay, here's an electron micrograph of E. coli. Simple prokaryotic organisms. They do not have the GFP gene. It's not part of their DNA. So these are the non-transformed bacteria. Here's what E. coli look like if you grow them on an agar plate. What are these little dots called? Remember from last semester? Did you guys look at plates at all last semester? Did you just have broth cultures?
No, we did the mouthwash experiment.
Okay, that's
was
right.
done with full plates.
Okay. So what are all these little dots on here? Okay. Yeah, they are colonies of E. coli. Is a colony an individual cell? No, I mean, E. coli are really, really tiny, aren't they? You can't see them barely even with your microscope on the oil immersion, the 1000x magnification lens. So these are masses of E. coli cells. There might be millions, hundreds of millions of E. coli making up that visible mass called the colony. The appearance of the colony, its color, its shape, the way its edges look, are all characteristics of the species of bacteria that make up that particular colony. So one of the things we're going to have you do is we're going to have you look at some plates that have E. coli colonies on them and make some observations about what do those colonies look like in the non-transformed bacteria. Okay? So kind of a baseline to see what does a normal E. coli colony look like. Now, we're going to work with some other plates, and these plates are going to contain a substance called ampicillin. What's ampicillin? It's an antibiotic, and what's it related to? It's related to penicillin. And what does penicillin do?
Fungus, isn't it?
Well, actually, penicillium is a organism, a fungal organism that can produce this particular antibiotic. But what does the antibiotic do? What does ampicillin do? It kills bacterial cells, okay? And it does so by interfering with their ability to make their cell wall. And if a bacterial cell can't make its cell wall, it's vulnerable and it dies. So if we take some of these non-transformed E. coli from this plate and transfer them onto a plate of ampicillin, what are we going to see? We're not going to see any colonies, right? Those cells won't grow. The ampicillin will kill the bacteria and inhibit the growth of any colonies. Okay, so what we want to try to do in the lab is to transform the normal E. coli cells and alter their genetic characteristics by inserting a foreign gene into these organisms. One of the genes that we're going to insert into them is a gene for the green fluorescent protein. Another gene we're going to insert into them is a gene for resistance to the antibiotic ampicillin. So if you have a resistance gene, can you grow on ampicillin? Yes, you can. If you don't have the resistance gene, you can't grow. So we're going to doubly transform our E. coli and make what we call a transgenic form of E. coli, something with some new DNA. So we're going to give it some new DNA. So how are we going to transform the E. coli? Well, we're going to use something called a plasmid. And you probably will talk a little bit more about plasmids in the lab session, right? But plasmids are little pieces of circular DNA. And the plasmid that we're going to use is a plasmid called P-GLO. P stands for plasmid. Okay? This is not a particularly big piece of DNA. It's a little circular piece of DNA that consists of 5,400 base pairs. Okay? What's a base pair? A base pair is like an AT or a GC. So it's not too big of a base pair. It's not too big of a piece of DNA. There are some important genes on this P-GLO plasmid. Okay? And when you think plasmid, think this is DNA. Okay? This is nucleotides. So here's our genetic information for the green fluorescent protein. This tells an organism this is how you make GFP protein, the stuff that fluoresces. This particular gene down here is the gene for ampicillin resistance. If you have this gene, then you can survive even if that antibiotic is present. This particular gene right here is a control element. And this control element is going to be important because it's going to help to regulate whether this green fluorescent protein gene is expressed. So it's kind of like an on-off switch. And this on-off switch is going to interact with something in the environment of the organism, a molecule called arabinose. Does anybody know what arabinose is? It's a sugar, good. Okay, glucose, OSC, arabinose, it's a different sugar. Okay. And if this particular sugar is present in the environment, then that sugar helps to turn on this on switch and allows then the GFP protein to be expressed and a green fluorescent protein to be produced using these instructions. If the sugar isn't present, then this switch doesn't get turned on and this gene doesn't get expressed, and so we can't produce the green fluorescent protein. Now, we'll talk later on after this upcoming exam when we talk about regulation of gene expression, how a molecule from the environment can turn on and turn off genes. Right now, for now, you're just going to have to accept the fact that arabinose reacts with this particular element of the P-GLO plasmid and basically serves as an on-off switch for gene expression. So, if we can infect normal E. coli cells with this P-GLO plasmid, now it should be able to tolerate the presence of ampicillin in its environment. It should be able to grow on ampicillin agar-containing plates. And if you also provide this bacteria with sugar, arabinose, in its environment, then the GFP protein should be expressed, and if you shine ultraviolet light on those cells, they should glow green. So here are some transgenic E. coli cells that have been transformed with the P-GLO plasmid. They've been provided the sugar that turns on the gene, and now they're expressing that GFP protein, and in the presence of the ultraviolet light, they're glowing green. And here's what they look like on an agar plate. If you shine ultraviolet light on those E. coli, the colonies actually glow green. Okay? Did they glow green prior to transformation? No. The normal, non-transformed E. coli don't have the genetic information to produce this protein. So this indicates a successful transformation of those E. coli. Okay, we already talked about this, the fact that there's this molecular switch that turns on the gene if the sugar is present. So, here's what transgenic E. coli look like on some of the media that you're going to be plating them out on during your lab experiment next week. Okay? LB basically is agar. Okay, it's a type of agar. So these are agar plates that contain ampicillin. These are agar plates that contain both ampicillin and the sugar arabidose. You see growth on both of those? So growth indicates that there are E. coli cells that are thriving and growing on those particular plates. So have these cells been successfully transformed? Yeah, because if they hadn't been transformed, if they hadn't taken up the plasmid, they would be killed by the ampicillin in the agar, and there would be no colonies. Are these successful transformants? Yes, they are, because not only are they growing, but those green fluorescent protein genes are being expressed because the sugar's turning on those genes in the plasmid. Okay? So both of these represent successful transformation of the E. coli cells. Green fluorescent protein has been genetically engineered not just into prokaryotic cells like E. coli, but there's been a fair amount of work that's been done to produce transgenic organisms in a number of different types of animals. Here is a green fluorescent protein pig, transgenic pig. There are transgenic mice. There's a green fluorescent protein bunny. And we can transform not only animals, but we can also transform yeast to have green fluorescent yeast. So this would be good St. Patrick's Day beer. So the ability to transform microorganisms by getting them to take up some foreign DNA from another source can have some pretty dramatic effects on altering their phenotype, making them resistant to an antibiotic or making them able to glow in the presence of ultraviolet light. What would you like to add in terms of background?
The purpose of developing these technologies wasn't to make things glow green. The value or the purpose of doing them with green fluorescent protein is GFP gave you a marker, gave you a way of knowing whether or not you got that gene into the cell. All right? It's not like scientists decided to, sit down and let's make green mice or green pigs. And, there's lots of different organisms that make green. The GFP was simply a tool to see if you could get a foreign gene into the cell, whether it be a bacterial cell or a eukaryotic cell, get it expressed, right? So it was a model, it was a tag, it gave us something to follow. The more important use of this technique is, for example, for gene therapy, if you would. Or, step back a little bit, modifying bacteria and yeast to produce genes that are useful industrially. For example, you probably, or your mom or dad, used laundry pre-treatments that take grass stains out of, a piece of clothing. You squirt the stuff on, you let it sit for a few minutes and it goes away. How does it do that? Well, somebody cloned a gene that cleaves the tail, the hydrophobic tail, off the chlorophyll molecule. Remember, the chlorophyll had this ring structure with magnesium in the middle and a long tail that anchored the chlorophyll into the membrane. Right? Chlorophyllase is the name of the enzyme. Clone the gene, you put it into a thick solution, usually with glycerol or polyethylene glycol, and then you spread it on the grass stain and you let it sit. Right? The gene goes in, cuts the tail off the chlorophyll molecule, and it no longer is green. Right? So this was taking a technique and applying it to a real-world problem to produce a product that was useful.
How many of you know someone who's diabetic? Lots of you, right? Okay. And if you're insulin dependent, okay, you need a source of insulin, right? The insulin that Lilly really made a lot of money on is an insulin called Humulin. That is human insulin. Human insulin is being produced by Lilly because they were able to isolate the human insulin gene and transfer it into a bacterial cell and grow up big vats of bacteria that had the human insulin gene. And they were able to produce insulin, and then Lilly was able to isolate the insulin from that growth media. Do you know where they got insulin before they did that? Does anybody know?
Where do you normally find insulin in the body? Pancreas.
Yeah.
All right. So where do you think we got insulin before we cloned the gene? Pancreas of humans, pigs, and cows. Normally it'd be thrown out or be incorporated into food for some other animal, but now they...
The problem with that is that pig insulin and cow insulin, if you inject that or ingest it, could be seen by your body as a foreign threat and a foreign molecule. And so some people had a reaction to those foreign forms of insulin. You don't have the same reaction to human insulin. And so human insulin had far fewer side effects. But it was all made possible by the fact that they were able to identify and isolate the gene and then transform E. coli cells with that particular genetic information and turn the bacteria into little insulin-making factories. So,
Yeah.
pretty cool technology.
Actually, they shifted from producing with bacteria to producing it in yeast, because yeast, remember, is a eukaryote. And what you will learn is that after a protein is produced, there are certain modifications that occur in the protein that happen in yeast that didn't happen in bacteria, and it produces a better, the glycosylation events occur.
One of the things that this particular lab will be good for is reinforcing a concept you learned last semester, and that is the importance of having proper controls in experimentation. And so one of the things you're going to be asked to do is to set up several different plates, and you will have to figure out which plate is serving as the experimental, and then which plate or plates are serving as controls for that particular experiment. Sometimes an experiment will have more than one control, and so there will be some questions that you will be asked about identifying control plates and what their function is.
Yeah, the protocol today is pretty simple and straightforward. You're doing a lot of things that are different than you've done before. This requires a good bit of imagination. You can't see the DNA. You can't see the bacteria. So you have to kind of think about what's going on in the tube in an abstract manner. So that'll be a bit of a challenge. With respect to controls, not experimental controls, but controls on experimentation. You mind if I take a few minutes?
No, go ahead.
This technique has been around for about 35 years. Back in the late 70s and early 80s, the ability to take pieces of DNA from one organism, put them into a vector, take that vector, put it into E. coli, and have the E. coli do fantastic things for us was just beginning to be developed. But scientists around the world realized there was a potential serious problem. E. coli was the workhorse. It's kind of like the mouse or the rat of the microbiological world. You have E. coli. Everybody's got E. coli in their intestines. Probably all mammals have E. coli in their intestines. And here we were going to take that organism and make it antibiotic resistant and produce things that normally it doesn't produce for our purposes. So scientists all over the world decided, wait a minute, we got to be careful with this thing. We're giving it antibiotic resistance. We're giving it new phenotypes. We got to be careful that it doesn't get out of the lab. So worldwide, in the early 1980s, scientists stopped doing this research because they realized there was a potential for some serious problems. And they got together at a conference center in California, a place called Asilomar, and sat down and over the course of, I think, a week or two, put together some guidelines as to how we were going to be able to do this research without endangering the population. And at first, some of the requirements were pretty draconian. For example, you had to have rooms with inside of rooms with inside of rooms, each of which was negative pressure to the one further inside, so that the opportunity for things escaping from the lab were much limited. I had the great fortune of building one of those labs in the biology building at the University of Notre Dame in the early 80s to handle encephalitis viruses, which created a similar issue. The consequence of all that and the creativity of the scientists doing this is that now what we have are E. coli cells, the ones that you're going to use, that have been engineered so that they cannot colonize your intestines. So now we can put antibiotic resistance into this microorganism. We can put these foreign genes in the microorganism, and we don't have to worry that if it gets out, that it will endanger the population. That doesn't mean you can be careless, right? But I think this is a really good example of how science can be very self-regulating, where scientists decide, well, we got to step back. This is potentially a real problem. What kinds of controls, not experimental controls, but controls on experimentation, we have to institute in order to do these things safely. And this type of consideration doesn't stop. When you think about the folks that handle Ebola patients, or now this whole issue with the Zika virus, which is going to be a big, interesting problem this summer, because the vector, the thing that transmits the virus from one individual to another, is a mosquito that's very common in the southern part of the United States. Scientists sit down and say, okay, what do we need to do to keep this under control? Right? It's great to be part of society that sits back and figures out how to do these sorts of things. Right? So because 30 years ago, scientists took the time to figure out how to prevent their experimental results from causing issues for the human society, we can do this now. We can do it in a high school lab. We can do it on the benchtop here in college with college freshmen, because that bacterium, E. coli K12, will not infect your intestines. Yes?
Couldn't it still mutate again, doesn't it?
There's always that risk.
There's always that risk, yeah.
There's always that risk. This lab is really pretty simple in conception, and like Dr. Morton said, it's routinely used in high schools as well. But it gives you some good fundamental background information, because we're going to take it several steps further in subsequent labs and do some more detailed application with it. So this is really kind of basic information you're going to learn this week.
And we're going to give you the plasmid that has the gene you're interested in transforming the bacteria. Beginning next week, you're going to take the gene and put it in the plasmid, take the plasmid, put it in the bacteria, grow out the bacteria, re-isolate the plasmid, and see which gene you put in it. All right, it's a bit more complicated. Correct me if I'm wrong, the final lab exam is entirely on these next 2 labs, correct?
Well, I mean, the remainder of the labs for the semester. I mean, it's
Right.
only 2 exercises.
Yeah. The 2 lab exercises we do this week and for the 4 weeks after this is what your entire final lab exam, or final exam, is on. So it's really important for you to understand the principles that we're addressing for each of the lab exercises for the next 5 weeks.
Okay, so you will get started on this week. Before we give you the reading quiz, any last-minute questions? All okay about DNA replication? Structure?